Dear students, the topic that I have selected for the online classes, that is microbes in human welfare. Actually, it is a 10th topic in your syllabus. Microbes in human welfare. If you observe these terms, microbes, human welfare, first we need to understand the term microbes. Since your primary school also we have studied microbes, but here first we need to understand the term like microbes as well as human welfare. First of all, what are microbes? Any living creature, any organism which is having less than zero point 0.1 millimeter size, they are called by the name microbes. Generally, their size is too small, as I have shown here, just 0.11 millimeter, less than that. Any creature that cannot be seen through our naked eye. Microscopes are required to see them means then you can call them as microbes. ಯಾವುದೇ ಆರ್ಗನಿಸಮ್ಸನ್ನು ಬರಿಗಣ್ಣಲ್ಲಿ ನೋಡೋದಕ್ಕೆ ಸಾಧ್ಯ ಇಲ್ಲ ಅಂತಂದರೆ ಅದನ್ನು ಮೈಕ್ರೋಬ್ಸ್ ಅಂತ ಕರೀತಾರೆ. Then, how many microbes are present within this world? Usually, the number of organisms which are present within this world, their exact number cannot be calculated. But according to one of the sources, believable sources, nearly about 1 trillion microorganisms are present within this world. But a detail regarding with all of them which is not available. ಒಂದು ಕ್ಯಾಲ್ಕುಲೇಷನ್ ಪ್ರಕಾರ ಸುಮಾರು 1 ಟ್ರಿಲಿಯನ್ ಅಷ್ಟು ಮೈಕ್ರೋ ಆರ್ಗನಿಸಮ್ಸ್ಗಳು ಇದ್ದಾವೆ. ಬಟ್ ಎಲ್ಲದ್ರ ಬಗ್ಗೆ ಡೀಟೇಲ್ ವಿಚ್ ಇಸ್ ನಾಟ್ ಅವೈಲೇಬಲ್. ದ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ಇಸ್ ವೇರ್ ಯು ಕ್ಯಾನ್ ಫೈಂಡ್ ದಮ್. ಹ್ಯಾಬಿಟಾಟ್ ಅಂತ ಕರೀತಾರೆ. ದಟ್ ಮೀನ್ಸ್ ವಿಚ್ ಕೈಂಡ್ ಆಫ್ ಎ ಪ್ಲೇಸಸ್ ದೇರ್ ಯು ಕ್ಯಾನ್ ಸಿ ದ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಮೈಕ್ರೋ ಆರ್ಗನಿಸಮ್ಸ್. ಫಾರ್ ದಿಸ್ ಆನ್ಸರ್ ಇಸ್ ದೇರ್ ಇಸ್ ನೋ ಫಿಕ್ಸ್ಡ್ ಪ್ಲೇಸ್ ಫಾರ್ ದ ಮೈಕ್ರೋಬ್ಸ್. In all possible places of this world, you can see the presence of microorganisms. Either the places where organisms can survive easily in such places, or in the places where condition is highly extreme, you can't expect the life of organism. In such places also it is possible to see the presence of microbes. Then what are those extreme places where you can see the presence of microbes? ಯಾವ ಕೆಟ್ಟ ಜಾಗಗಳಲ್ಲೂ ಅದು ಬದುಕುತ್ತೆ ಅಂತ ಕೇಳಿದ್ರೆ, where salinity is too high or where temperature is too high, that means even temperature which is exceeding more than 100 degrees centigrade too. We will feel uncomfortable even temperature is nearly about 36 or 38, especially during hot summer. ಬೇಸಿಗೆ ಕಾಲದಲ್ಲಿ 40 ಡಿಗ್ರಿನೇ ನಮಗೆ ಸಾಧ್ಯ ಇಲ್ಲ ಅಂತದ್ರಲ್ಲಿ ಈವನ್ ಸಮ್ ಆಫ್ ದ ಮೈಕ್ರೋ ಆರ್ಗನಿಸಮ್ ಈವನ್ ದೇ ಕ್ಯಾನ್ ಟಾಲರೇಟ್ ಅಪ್ ಟು 100 ಡಿಗ್ರಿ ಸೆಂಟಿಗ್ರೇಡ್. ಇನ್ ಸಚ್ ಪ್ಲೇಸಸ್ ಆಲ್ಸೋ ಯು ಕ್ಯಾನ್ ಸಿ ದ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಮೈಕ್ರೋಬ್ಸ್. ಈವನ್ ಇನ್ ದ ಪ್ಲೇಸಸ್ ವೇರ್ ಅಸಿಡಿಟಿ ಇಸ್ ಟು ಹೈ, ದಟ್ ಮೀನ್ಸ್ ಆಸಿಡ್ ಕಂಟೆಂಟ್ ಇಸ್ ಟು ಹೈ, ಇನ್ ಸಚ್ ಪ್ಲೇಸಸ್ ಆಲ್ಸೋ, ಆರ್ ಇನ್ ದ ಪ್ಲೇಸಸ್ ವೇರ್ ಹೈ ಸ್ನೋಫಾಲ್ ವಿಲ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್. ಹಿಮ ಜಾಸ್ತಿ ಎಲ್ಲಿ ಬೀಳುತ್ತೋ ಅಂಥ ಜಾಗದಲ್ಲೂ ಕೂಡ microorganisms ಇದಾವೆ. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ಹಿಯರ್ ಈಸ್ ಯಾವೆಲ್ಲ ಆರ್ಗನಿಸಮ್ಸ್ ನ ಮೈಕ್ರೋಬ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ವಾಟ್ ಆರ್ ಆಲ್ ದ ಡಿಫರೆಂಟ್ ಕ್ರಿಯೇಚರ್ಸ್ ದಟ್ ವಿಲ್ ಬಿ ಕಾಲ್ಡ್ ಬೈ ದ ನೇಮ್ microbes, which are considered as microbes? As we have studied earlier itself, virus, bacteria, fungus, protozoans, blue-green algae. These are the few examples for the microbes. The virus, bacteria, fungus, protozoans, and even blue-green algae. Otherwise, you can call this as cyanobacteria too. First year itself, we have studied about them. Virus, which is not belonging to any of the kingdom because of its dual nature. Bacteria belonging to monocyte cell. Fungus belonging to mycota. Protozoans, they are the member of protista. Then blue-green algae, nothing but cyanobacteria, again belonging to monocyte self. These creatures, their size is highly limited. Their observation is possible only with the help of microscope. They are called by the name microbes. Generally, microbes means, as we have studied in our previous classes, we will consider them that pathogens. Most of the microbes, as we have studied, they are pathogens. They will cause various type of diseases. That means in our mind, we have fixed ourselves that microbes are harmful. They will cause diseases. ನಮ್ಮ ತಲೆಯಲ್ಲಿ ಏನು ಫಿಕ್ಸ್ ಆಗಿದೆ ಅಂದರೆ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ ಅಂತಂದರೆ ದೇ ವಿಲ್ ಕಾಸ್ ಡಿಸೀಸಸ್ ಅಂತ ಹೇಳಿ. ಆದರೆ all the microorganisms, they are not pathogens. Many of the microbe, even they are advantages for us. They are useful in case of human welfare. In this topic in second year, just we will study about advantages of microbes in human welfare. Next, microbial culture, the next term that we need to study. ಏನು ಹಾಗಂದ್ರೆ ಮೈಕ್ರೋಬಿಯಲ್ ಕಲ್ಚರ್ ಅಂತಂದ್ರೆ microorganism, they will grow in various places as we have discussed earlier. But next question is whether it is possible to grow microbes under laboratory conditions. ಅಂದರೆ ಲ್ಯಾಬೊರೇಟರಿ ಕಂಡೀಷನ್ಸ್ ಅಲ್ಲಿ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಸ್ ಅನ್ನ ಗ್ರೋ ಮಾಡೋದಕ್ಕೆ ಸಾಧ್ಯನಾ? whether it is possible to grow or culture the microbe within our laboratory conditions. Yes, it is possible to grow them, but how? By using cultural vessels. Any glass vessels like test tube or petri plate or beaker or conical flask that you can take for their culture. Just you need to provide them nutrient media for them. ನಾವು ಹೇಗೆ ಊಟ ಮಾಡ್ತೀವೋ, ನಮ್ಗೆ ಹೇಗೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಬೇಕೋ ಹಾಗೇನೆ ಮೈಕ್ರೋಬ್ಸನ್ನು ಲ್ಯಾಬ್ರೇಟರಿಲಿ ಬೆಳೆಸೋದಾದರೆ ಈವನ್ ಇಟ್ ರಿಕ್ವೈರ್ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್. ದೋಸ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಆರ್ ಟು ಬಿ ಟೇಕನ್ ವಿತಿನ್ ದೋಸ್ ಕಲ್ಚರಲ್ ವೆಸಲ್ಸ್. ನೀವೇನು ತಗೊಂಡಿರ್ತೀರೋ ಅದು ಗ್ಲಾಸ್ ಟ್ಯೂಬ್ ಆಗಿರಲಿ, ಪೆಟ್ರಿ ಪ್ಲೇಟ್ ಆಗಿರಲಿ, ಬೀಕರ್ ಆಗಿರಲಿ, ಕೋನಿಕಲ್ ಫ್ಲಾಸ್ಕ್ ಆಗಿರಲಿ, ಅದರಲ್ಲಿ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಮ್ನ ಹಾಕಿ if you maintain entire sterilized condition, aseptic condition, it is possible to grow the microorganisms including bacteria or fungus too. How you can see them? Once they were grown within cultural vessels, by utilizing the nutrient media, they will form colonies or groups. ಹೌದು ಬೆಳೆಸಿದೀವಿ, ಬಟ್ ನಮ್ಗೆ ಹೆಂಗೆ ಗೊತ್ತಾಗುತ್ತೆ, ಕಾಣಿಸುತ್ತಾ ಅಂತ ಕೇಳಿದ್ರೆ, you can see the presence of colonies within cultural vessels once microbes were grown. ಒಮ್ಮೆ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಸ್, ಅದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಆಗಿರಲಿ, ಫಂಗಸ್ ಆಗಿರಲಿ ಬೆಳೆದು ಅಂತಂದರೆ ಆ ಕಲ್ಚರಲ್ ವೆಸಲ್ಸಲ್ಲಿ ನಿಮಗೆ ಅದರ ಗ್ರೂಪ್ಸ್ ಅಂದರೆ ಕಾಲೋನೀಸ್ ನಿಮಗೆ ಕಾಣಿಸುತ್ತೆ. ಸಮರಿ ಏನಪ್ಪ ಅಂತಂದರೆ, ಈವನ್ ಇಟ್ ಈಸ್ ಪಾಸಿಬಲ್ ಟು ಗ್ರೋ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಸ್ ಬೈ ಪ್ರೊವೈಡಿಂಗ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಮ್. ಇಟ್ ಈಸ್ ಪಾಸಿಬಲ್ ಟು ಗ್ರೋ ದೆಮ್ ವಿತಿನ್ ಕಲ್ಚರಲ್ ವೆಸಲ್ಸ್ if you maintain sterilized aseptic condition within your laboratories. The next one, what are all the different advantages of microbes? Microbes in the ಏನು advantages ಇದೆ ನಮಗೆ, human being ಏನು advantages ಇದೆ ಅಂತಂದ್ರೆ, in this topic we will study about various advantages of microorganisms. First of all, in our daily routine activities, how microbes are useful? Is there any involvement of microorganism in preparation of household product? That we will study. Then how microorganisms are useful in case of preparation of antibiotics? How microbes are needed for sewage water treatment? How microbes are useful in biogas preparation? How microbes are used in preparation of bioactive molecules? These informations will be studied one by one here. ಅದು ಯಾವ್ದ್ಯಾವುದು ಅಂತ ಒನ್ ಬೈ ಒನ್ ಹೇಳ್ತಾ ಹೋಗ್ತೀವಿ. ಅದರಲ್ಲಿ ಮೊದಲನೇದು, ಹೌ ಮೈಕ್ರೋಬ್ಸ್ ಆರ್ ಯೂಸ್ಫುಲ್ ಇನ್ ಪ್ರಿಪರೇಷನ್ ಆಫ್ ಹೌಸ್ ಹೋಲ್ಡ್ ಪ್ರಾಡಕ್ಟ್. ದಿನನಿತ್ಯ ನಾವು ಸೇವಿಸುವಂಥ ಆಹಾರದಲ್ಲಿ ಹೇಗೆ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಗಳ ಇನ್ವಾಲ್ವ್ಮೆಂಟ್ ಇದೆ. ಹೌ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಸ್ ಆರ್ ನೀಡೆಡ್ ಫಾರ್ ಪ್ರಿಪರೇಷನ್ ಆಫ್ ಸಮ್ ಆಫ್ ದ ಡೈಲಿ ರೊಟೀನ್ ಫುಡ್ ವಾಟ್ ವಿ ಆರ್ ಕನ್ಸ್ಯೂಮಿಂಗ್. ವಾಟ್ ಆರ್ ದೋಸ್? either in preparation of curd or in preparation of some dishes like dosa or idli, or preparation of toddy, and preparation of soya product, and even for preparation of cheese and preparation of bread. These are all various food preparation which is needed for our daily life. Most of them, how microbes are involved in this. This information will be given by one. These are all the products which are used by us in our daily life, and how microbes are useful for preparation of these products which will be explained here. Okay, let's see here. As I have mentioned earlier, the product which we will be using in our daily life, how microbes are important. What are the product? Which is the microbes? What is its role? That we will explain here. Let's see one by one. First one, curd. That is food, most important one, and we are using them regularly in our daily life, including me, including yourselves, all of us, we are using this particular milk product. Fermentation of the milk will results in the formation of curd. How curd will be prepared? Which activity is involved? Which microbe is involved? That we will see here. First of all, the preparation of the curd. ಮೊಸರು ಕಾಮನ್ ಆಗಿ ಗೊತ್ತಿರುವಂಥದ್ದೇ, ಡೈಲಿ ಲೈಫಲ್ಲಿ ಯೂಸ್ ಮಾಡುವಂಥದ್ದು. ಹಾಲಿಂದ ಮೊಸರಾಗತ್ತೆ ಅಂತ ಗೊತ್ತು, ಹೇಗೆ ಯಾಕೆ ಅನ್ನೋದನ್ನು ಕಲಿಸ್ಕೊಡ್ತಿದ್ದೀವಿ ಅಷ್ಟೇ. ಫಸ್ಟ್ ಆಫ್ ಆಲ್, ಹೌ curd will be prepared. The method which is involved in preparation of the curd. First, milk that will be boiled, then it will be kept for cooling less than 40 degrees centigrade. You need to cool them up to 40 degrees centigrade or less than that. Then addition of previous day curd as inoculum. ಅಂದರೆ ಹಿಂದಿನ ದಿನದ ಮೊಸರನ್ನು ಒಂದು ಚಮಚವನ್ನು ನೀವು ಕೂಲ್ ಮಾಡಿರುವಂಥ ಹಾಲಿಗೆ ಮಿಕ್ಸ್ ಮಾಡ್ತೀರಿ. The procedure which is involved in preparation of the curd is, first we need to boil the milk, which is to be cooled up to 40 degree or less than that. You need to add 1 tablespoon of previous day curd as inoculum. Then you need to mix that. If you mix that and if you keep them undisturbed for few hours, 6 to 8 hours, then there will be conversion of milk into curd. The process which is involved, which is called by the name fermentation, catalyzed by or the performance of the conversion that was done by Lactobacillus acidophilus. The inoculum that we have added, that means previous day curd that we have added, it is consisting a bacteria called by the name Lactobacillus acidophilus. Then what is the role of this? What is the role of Lactobacillus acidophilus? How it can convert milk into curd? Let's see together here. First of all, this Lactobacillus acidophilus, which will release an enzyme called by the name lactase. ಏನು ಮಾಡುತ್ತೆ ಲ್ಯಾಕ್ಟೋಬ್ಯಾಸಿಲಸ್ ಅಸಿಡೋಫಿಲಸ್? ಎಲ್ಲಿರುತ್ತೆ? ನೀವು ಹಾಲನ್ನು ಬಾಯ್ಲ್ ಮಾಡಿದ್ರಿ, ಕೂಲ್ ಮಾಡಿದ್ರಿ, 1 ಟೇಬಲ್ ಸ್ಪೂನ್ ಅಷ್ಟು ಕರ್ಡನ್ನು ಆ್ಯಡ್ ಮಾಡಿದ್ರಿ. ಆ ಕರ್ಡಲ್ಲಿ ಇದ್ದಂಥ ಲ್ಯಾಕ್ಟೋಬ್ಯಾಸಿಲಸ್ ಅಸಿಡೋಫಿಲಸ್ ವಿಚ್ ವಿಲ್ ರಿಲೀಸ್ ಅನ್ ಎನ್ಸೈಮ್ ಕಾಲ್ಡ್ ಬೈ ದ ನೇಮ್ lactase. This lactase which will act on milk sugar, carbohydrate present within the milk, that is called by the name lactose. This lactase will act on this milk sugar, lactose, and convert this lactose into lactic acid. What are the event? First of all, this Lactobacillus acidophilus, it has released an enzyme called by the name lactase, which acted upon milk sugar lactose, and it has converted lactose into lactic acid. Then this lactic acid which will coagulate, which will clump milk protein called by the name casein to convert finally milk into curd. ಏನಾಗಿದೆ ಇವೆಂಟ್ ಒಮ್ಮೆ ನೋಡೋದಾದ್ರೆ, ಲ್ಯಾಕ್ಟೋಬ್ಯಾಸಿಲಸ್ ಅಸಿಡೋಫಿಲಸ್ ಆ ಕರ್ಡ್ ಟೇಬಲ್ ಸ್ಪೂನ್ ಅಲ್ಲಿ ಇತ್ತು, ಅದು ಲ್ಯಾಕ್ಟೇಸ್ ಅನ್ನೋ ಎಂಜೈಮನ್ನು ರಿಲೀಸ್ ಮಾಡಿದೆ. ಆ ಲ್ಯಾಕ್ಟೇಸ್ ಅನ್ನೋ ಎಂಜೈಮ್ ಮಿಲ್ಕ್ ಶುಗರ್ ಲ್ಯಾಕ್ಟೋಸನ್ನು ಲ್ಯಾಕ್ಟಿಕ್ ಆಸಿಡ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಮಾಡಿದೆ. ದಿಸ್ ಲ್ಯಾಕ್ಟಿಕ್ ಆಸಿಡ್ coagulate the milk protein called by the name casein to form curd finally. That means milk consisting of both carbohydrate as well as protein. Carbohydrate, it was present here, it was called by the name lactose. Then protein which was present, that is casein. Lactobacillus acidophilus, it has released lactase. This lactase acted upon lactose. Lactose was converted into lactic acid. Then this lactic acid coagulate that milk protein called by the name casein to form finally curd. That means the role of Lactobacillus acidophilus in the conversion of milk into curd, that can be noticed from this. Then this Lactobacillus acidophilus can be considered as lab, lactic acid bacteria. The reason is well known here, because here due to activity of Lactobacillus acidophilus only, there was a release of lactic acid. Then the one important word here, probiotics, which is to be known. What are probiotics then? Any live microbe, any live bacteria or any live fungus that we have consumed along with our food, if they can improve our mental or physical health, then you can call that as probiotics. ಯಾವುದೇ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಯಾವುದೇ ಫಂಗಸ್ಸನ್ನು ಆಹಾರದ ರೂಪದಲ್ಲಿ ನೀವು ತಗೊಂಡ್ರೆ ಅದು ನಿಮ್ಮ ದೇಹದ ಹೆಲ್ತನ್ನು ಕಾಪಾಡುತ್ತೆ ಅನ್ನೋದಾದರೆ, ಐದರ್ ಇಟ್ ಮೇ ಬಿ ಫಿಸಿಕಲ್ ಹೆಲ್ತ್ ಆರ್ ಇಟ್ ಮೇ ಬಿ ಮೆಂಟಲ್ ಹೆಲ್ತ್, ದೆನ್ ಇಟ್ ಕ್ಯಾನ್ ಬಿ ಕಾಲ್ಡ್ ಬೈ ದ ನೇಮ್ probiotics. Consumption of the curd, which will give two important benefit for us. What are those? First of all, this Lactobacillus acidophilus or lactic acid bacteria, it will increase the content of vitamin B12 within curd. ಹಾಲು ಮೊಸರಾಗುವಂಥ ಆ ಪ್ರಕ್ರಿಯೆಯಲ್ಲಿ ಆ ಮೊಸರಲ್ಲಿ ವಿಟಮಿನ್ B12 ಕಂಟೆಂಟನ್ನು ಜಾಸ್ತಿ ಮಾಡುತ್ತಂತೆ ದಿಸ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. And another importance of this, that is prevention of disease causing bacterial growth in case of our stomach. Regular usage of curd that can prevent growth of disease causing bacteria within our digestive system, within our stomach, within our gut. This is the importance of lactic acid bacteria, otherwise you can consider that as probiotics. The question may be like this: mention the importance of lactobacillus, or mention the importance of lactic acid bacteria. First of all, they will improve the content of vitamin B12. Then second importance is, if this curd is consumed, it consisting Lactobacillus acidophilus, it can prevent the growth of disease causing bacteria within our digestive system. This is all about curd and its importance. Okay, after completion of first important fermentative food product of the milk, it was curd, that already I have explained. The second one, that is related with one of the famous important daily routine breakfast, that is related with idli and dosa preparation. As we know that especially in south part of the India, this is the regular breakfast food for us. In most of the houses, including your houses too, idli and dosa, that is said to be most regular food of us. Whether here also there is an involvement of microbe for its preparation means, yes, again this process of preparation of these two breakfasts, it is needed, it is possible only through the activity of bacteria like Konnurs mesenteroid Streptococcus faecalis. ಹೇಗೆ ಮಾಡ್ತಾರೆ ಇದನ್ನು ಅಂತ ನೀವು ನೋಡೋದಾದ್ರೆ, as we know that black gram and rice, that will be grinded either by using mixer or grinder. This black gram and rice, that will be kept for grinding. Once it was grinded, actually the texture will be rough. When it was grinded, the structure of this, it will be rough. But same batter, if you keep that for at least 6 to 8 hours or more than that, next day if you observe, then the same batter, it will look very smooth. How it is possible? The same batter, when it was prepared previous day by using black gram and rice, it was little bit rough, but it was kept for 8 hours or 6 hours or more than that, that nature of the batter, it will be smooth. This conversion is possible only due to activity of bacteria like Konnurs mesenteroid Streptococcus faecalis. Mechanism is called by the name ಡಫ್ಲಿಂಗ್ ಅಂತ ಕರೀತಾರೆ. ಕನ್ನಡದಲ್ಲಿ ಇದನ್ನು ಹುದುಗುವಿಕೆ ಅಂತ ಕರಿತೀವಿ. The process is simple. Black gram and rice, it was grinded. After grinding them, the texture of that batter, it will be rough. it will be kept for overnight, at least 6 to 8 or 10 hours. Then the next day, if you observe, the nature of the batter will be smooth. This is possible only due to activity of microbes again. That means for second important preparation also there is the involvement of microbe. For conversion of milk into curd, Lactobacillus acidophilus, conversion of rough batter of black gram and rice, again for the preparation of breakfast like idli and dosa, that bacteria which was involved here, Leuconostoc mesenteroid and Streptococcus faecalis. Next, one of the energy drink that will be used in case of many part of India including south part too, that is called by the name toddy. How toddy will be prepared? By using coconut milk. ಎಳನೀರು ಅಂತ ಏನು ಕರಿತೀರಿ, ಅದರ ಮಿಲ್ಕ್ ಕಂಟೆಂಟ್ ಏನಿರುತ್ತೆ ಅಥವಾ ಎಳನೀರು ಅಂತ ಏನು ಕರಿತೀರಿ, ಅದನ್ನ ಫರ್ಮೆಂಟೇಷನ್ ಆಗೋದಕ್ಕೆ ಬಿಡ್ತಾರೆ. The coconut milk that will be taken from the coconut or palm tree that will be kept for fermentation. Then this toddy will be prepared, that will be used as energy drink in many part of India including south part too in some places. Then some of the soya product, soya bean, I think you might have seen, you are using that as a food. In that soya bean, many of the product will be produced again due to activity of microbes itself. One of the Indonesian food called by the name tempeh, that will be prepared by using fungus like Rhizopus and mucor. They were not bacteria, they were the fungus, but it will be used for the preparation of this particular food product, especially in case of Indonesia, called by the name tempeh. Then tofu, which is cheese like product, that will be produced by using fungus again, that is mucor. Then soya sauce, that will be used in case of most of the North Indian food items including fried rice or including Manchurian. That soya sauce that we will be using, again it is a product from soya bean, that will be used again through action called by the name fermentation. That action of fermentation, that will be, there will be changes in chemical nature of the substances, it will be converted into some other form. That was possible here because of activity of some fungus. That means even for the preparation of variety of soya product, microbes like fungus that was utilized. Then bread, bakery item which is most familiar, most popular, most of the people in case of all houses, one or other way we will be using the bread items, either directly or by roasting them or sandwiching them or by using that in the form of burger etc. This bread will be used. How this bread will be prepared? Just we know the taste of them, that is delicious, but how it will be prepared? By using wheat flour or maida, all purpose flour anta karitare, that will be mixed with baker's yeast, Saccharomyces cerevisiae, which is a fungus, that will be mixed. Yeast granules anta sigate, ಅದನ್ನು little bit of water ಅಲ್ಲಿ ಇದನ್ನು ಡಿಸಾಲ್ವ್ ಮಾಡ್ತಾರೆ, ವೀಟ್ ಫ್ಲೋರ್ ಜೊತೆಗೆ ಅದನ್ನು ಮಿಕ್ಸ್ ಮಾಡ್ತಾರೆ, ಆ ಕಂಟೆಂಟನ್ನು ಹಾಗೆ ಇಡ್ತಾರೆ. ಫಸ್ಟ್ ಎಷ್ಟು ಫ್ಲೋರ್ ಇರುತ್ತೋ ಅದಕ್ಕಿಂತನೂ ಜಾಸ್ತಿ ಆಗುತ್ತೆ ವಾಲ್ಯೂಮಲ್ಲಿ ಬಿಕಾಸ್ ಆಫ್ ಆಕ್ಟಿವಿಟಿ ಡನ್ ಬೈ ಈಸ್ಟ್. Next it will be kept for baking. Then you can see at finally the bread slice which is very soft. How it was possible? You have mixed wheat flour with yeast granule, then you have kept for some hours, then you have baked them, then you have obtained bread slices which is having smoothness. This softness is due to release of carbon dioxide. This carbon dioxide gas will be released during the time of fermentation, catalyzed, or that was done by Saccharomyces cerevisiae, which is generally known as baker's yeast here, which is nothing but a fungus. That means for the preparation of bread also here microbe, that is nothing but yeast, Saccharomyces cerevisiae, it was used. For curd preparation, Lactobacillus acidophilus. For duffling, Leuconostoc mesenteroid and Streptococcus faecalis. For soya product, Rhizopus and mucor. It was used for the preparation of bread. Here, Saccharomyces cerevisiae, that is nothing but yeast. It was used. Then, cheese. I think even you are familiar with this. You might have seen this either in the form of cube or in the form of slices. Cheese, I think many of you have seen. ಈ ಬರ್ಗರ್ನ ಮಾಡಬೇಕಾದ್ರೆ ಅದನ್ನು ಲೇಯರ್ ಆಗಿಡ್ತಾರೆ, ಸ್ಯಾಂಡ್ವಿಚ್ ಮಾಡಬೇಕಾದ್ರೆ ಚೀಸನ್ನು ಹಾಕ್ತಾರೆ, ಪಿಜ್ಜಾ ಅಂತ ಪ್ರಿಪ್ರೇಷನ್ನು ಚೀಸ್ ಇಲ್ಲದೆ ನಡೆಯೋದೇ ಇಲ್ಲ. ಆ ಚೀಸ್ ಪ್ರಿಪ್ರೇಷನ್ ಹೇಗಾಗುತ್ತೆ, ಅದರಲ್ಲಿ ಏನಾದರೂ ಮೈಕ್ರೋಬ್ಸ್ ಇನ್ವಾಲ್ವ್ಮೆಂಟ್ ಇದೆಯಾ ಅದನ್ನು ನೋಡಬೇಕು. In your theory syllabus, they have given about two different type of cheese product. One is Swiss cheese, another one is Roquefort cheese. Swiss cheese that will be prepared due to activity of one of the bacteria called by the name Propionibacterium sharmani. Propionibacterium sharmani, which is the name of the bacteria that will be utilized for the preparation of Swiss cheese. ಅಲ್ಲ ಎಲ್ಲ ಬಿಟ್ಟು ಸ್ವಿಸ್ ಚೀಸೇ ಯಾಕೆ ಅಂತ ಕೇಳಿದ್ರೆ, this Swiss cheese which is famous for its texture, which is famous for its flavor, and which is famous for its taste. What about texture? This Swiss cheese which is having large holes. This large holes in case of Swiss cheese, this is due to release of carbon dioxide, again due to activity of Propionibacterium sharmani. Propionibacterium sharmani, this is said to be a bacteria that is used for the preparation of Swiss cheese. Why Swiss cheese is famous? For its texture which is having large holes, for its flavor, for its taste too. Then another type of cheese that is called by the name Roquefort cheese. Here this type of cheese generally that will be prepared by using sheep's milk. For this preparation, fungus called by the name Penicillium roqueforti that will be used. That means even for the preparation of cheese, there is the involvement of some of the microbes. That means we have explained various food products like curd or second one, idli and dosa preparation, for soya product, for toddy preparation, for bread preparation, even for the cheese preparation without microorganism, which is highly impossible. Okay, dear friends, so far I have explained various concepts regarding with microbes in human welfare. First, I have told about microbe, this organism which is having less than 0.1 millimeter, which cannot be seen through naked eye. They are generally called by the name microbes. They are present in all possible places, either within water, within air, within soil, in all possible places, even in the places where life is not easy. In such places also they can survive at high salinity, at high temperature, or in case of high acidity. In such worst environmental condition also they can easily survive. Generally virus, bacteria, fungus, protozoans, cyanobacteria, they can be called by the name microbes. They can be grown artificially by providing nutrient medium under aseptic sterilized condition by using cultural vessels. Many of food preparation that need involvement of microbes. For example, curd preparation, for that Lactobacillus acidophilus, which will release lactase. Lactase will convert lactose into lactic acid. Lactic acid will coagulate milk protein to form curd. Then breakfast like idli and dosa, black gram will be mixed with rice. It will be grinded. First batter will be thick, then it will be smooth because of two important bacteria, Leuconostoc mesenteroid and Streptococcus faecalis. Then toddy, energy drink in case of most part of the India, including south part of the India too. That will be prepared by using coconut water through the activity like fermentation. Then another important preparation we have explained that is related with bread preparation. For that yeast it was utilized. Yeast will be used along with wheat flour, and after that smoothness it was because of release of carbon dioxide that I have explained. Then Swiss cheese and Roquefort cheese. Two important varieties of cheese I have told. Swiss cheese by using Propionibacterium sharmani. It is known for flavor, taste and texture. Texture, that is nothing but large holes. In case of Swiss cheese, it is because of Propionibacterium sharmani. Then cheese, Roquefort cheese, that is due to involvement of Penicillium roqueforti, that particular fungus, by using sheep milk. Then some of the soya products I have explained, either it may be tempeh or it may be tofu or soya sauce, especially by using fungus like Rhizophus and mucor. These food preparation and what kind of a bacteria, which fungus that was utilized, that I have explained in this particular video class. I have given some question here. The first question for your assignment, name the bacterium used for Swiss cheese production, name the microbe which is used for bread preparation, and mention the importance of lactic acid bacteria. I have told you to remember usually lactic acid bacteria that will improve vitamin B12 content within the curd. Second one, they will prevent unwanted growth of disease causing bacteria within our stomach. These questions I have given. You can solve this question and you can send that answer for this question through WhatsApp.